Good afternoon, everyone. Welcome to our fifth UConn Health and Jackson Laboratory Postdoc Research Day. I'm so glad all of you could join us as we celebrate National Postdoc Appreciation Week with a sample of our excellent postdocs here. I'm sorry that we have to be over a screen again this year, but it is what it is. We'll make the most of it. I'll let you know we've hired some professionals this year to help manage this event so that we have a much smoother event than we had last year. And I'll talk more about that in just a second. But first, next, please, some thank yous. Next. There we go. Some thank yous. As always, I want to thank the leadership of the UConn School of Medicine and School of Dental Medicine for supporting us in this event, and in particular, the Health Center Research Advisory Council for the financial support that made today possible. A big thank you to Stephanie Holden for all of her work in arranging and organizing this event, as well as Diane Starkoziak. Our partners at Jackson Lab were Dr. Sarah Wojcicki and Katie Savigny, so thank you to them as well. And we're working here today with Venue Audiovisuals, who is managing our event, as well as our poster session tomorrow. And that is Chris Watson and Mason, who's our technical support today. I also want to give a big thank you to the three enthusiastic postdocs, Shirley, Roberto, and Feza, who made up our organizing committee this year. They helped sort of with all of the planning to design this couple of days and make it possible, put everything together in terms of schedules and program books. And so they did a great job. And thank you to them. And you'll hear from all three of them in just a moment. And our talks today it's sort of a competition. There's a little bit of prize money at stake here for our speakers this afternoon. And I want to thank Doctors Garrett, Levine, and Lee for agreeing to spend the afternoon with us and judging our day for that purpose. So if you haven't been to our event in years past, I think you're in for a treat. The way we do this is you're going to hear from 27 different postdocs from around the UConn Health and Jackson Labs campus here in Farmington. They're each going to have 4 minutes and a single slide to try to convince you that their work is the coolest thing on the planet. And it's great. It's a really fun day. You get to hear a wide swath from a broad array of scientists on the research that's going on here on our campus. So I think it's a great event. This will continue in tomorrow. So next, please. Where we will have a virtual poster session tomorrow at 3 o'clock. There are two sessions, one from 3 to 4 and one from 4 to 5. You can see on the screen in front of you, we've broken our postdoc presenters into these groups, and you can find your favorite postdoc and come out and support them. That will also be through the same platform. So the link that you use to get into the talks today also has a button for the poster session tomorrow. So same link. The program book should also be there as well. I just want to talk really quickly about what's going to happen tomorrow to prepare everybody a little bit. So next, please, if you would. When you come in tomorrow and you click on the poster session link, you'll come to sort of a lobby, an artificial looking lobby on your screen, and you will control a little avatar. So you can see at the top of this page is the avatar for Chris, our venue host, and you can pick on the right-hand side the different group that you want to go see. You can go around and visit all 8 groups if you want. And when you do, you click on group 1, for example, you'll enter the room, and this is what it'll look like when you enter. You'll see the 4 posters in this group. Your avatar will be at the top. What you don't see now, but what you will see tomorrow, are the avatars for the presenters. They should be standing somewhere near their poster in this virtual space. And so if you want to go see, for example, Hassan's poster, you would drag your avatar over next to him. And as you get close to him, you'll begin to see and hear him as well. And if you've got a webcam and you got your microphone on, you can have a back and forth conversation. And anybody who's in the neighborhood, you'll be able to hear. So just like at a poster, if there's 2 or 3 of you standing around talking about a poster, you'll all be able to do that. So I think it'll be a really nice virtual session that'll be a lot more interactive than what we were able to do last year. As you can see, if you are focusing on Hassan's poster and you actually want to see it, you can, of course, zoom in. So you can use your mouse or your keyboard, or in the bottom right-hand corner, there's a little controller that you can zoom in so that you can focus on different aspects of an individual poster on this page. So hopefully that works really well. I think that'll be a lot of fun tomorrow. But in the meantime, let's get back to our talks. And so with that, enough from me. Let's begin our day with our first speaker. Thank you for coming. Enjoy, everybody.
Hello, I am Antoine McFarland, and recent discoveries link age-related decline of all cell types to the accumulation of senescent cells. Our project is centered on bone repair by mediation of senescent cells via timed drug release from calcium phosphate disc, specifically for elderly women. To understand why, let me first define senescent cells. Next. They are cells that have undergone a change, next, due to advanced telomere erosion, other DNA damage such as double-strand breaks, or factors such as oxidative stress causing them to lose one or more normal cell function, but mainly cell division. Next. Additionally, senescent cells are larger than normal cells. Next. And they secrete cytokines and chemokines that are normally not secreted in levels of that quantity by normal cells. The secretion of these cytokines and chemokines is tied to senescence associate secretory phenotype, or SAS for short, which stimulates chronic inflammation. Next. Chronic inflammation associated with senescent cells accumulation can add to, next, slowed bone healing for elderly patients. Next, the inflammation inhibits macrophage phenotypical change from pro-inflammatory M1 to anti-inflammatory M2 phenotypes that resolves inflammation and guides bone formation. Slowed bone healing in elderly is problematic, and a crucial solution is needed to improve elderly bone healing. Longer bone healing times in these elderly patients and the subsequent inactivity is detrimental to their long-term health. We devised two approaches to mitigate the SAS using one of two drugs. Firstly, senescent cells chronic inflammatory effects can be downregulated by using anti-cancer drugs known as senolytics, such as Novaclax or ABT-263 for short, to selectively trigger senescent cell death. Next. Secondly, anti-inflammatory drugs such as ruxolitinib, aka Rux, which specifically inhibits JAK pathways, blocks the destructive pro-inflammatory SAS secretions. Next. However, preliminary data revealed substantial evidence that the time window for drug delivery is crucial for ensuring positive effects and reducing negative effects on drug healing. We hypothesized that the senolytic drug ABT-263 or the anti-SAS drug Rux can positively affect bone healing, but they must be optimized in their delivery profiles to align with their effects on senescent cells and cells involved in bone healing. Next. Past experiments done in our lab have shown that we can load drugs under a calcium phosphate transient barrier layer, known as TBL, and delay their release for 2 to 3 days post-implantation. By utilizing drug loaded beneath the transient barrier layer disc that have a timed release, we anticipate that our two strategies could provide a method for treating slow bone healing in the elderly. Thank you for your time.
Hi everyone, I'm Wenchen Du. I'm a postdoc from Kishi's laboratory at UConn Health Department of Biomedical Engineering. So today I'm going to share with you a disease called Plancita acreta spectrum. So Plancita is an organ that connects fetus and the mother. It is critical to the survival and the development of the fetus. To provide sufficient nutrition, Plancita needs to invade into the maternal endometrium. Endometrium produces factors that promote Plancita invasion and inhibits overly aggressive invasion. However, in some conditions, the Plancita invades abnormally deep into the wall of myometrium layer of the uterus, or even deeper. This series of abnormal Plancita invasion is called Plancita acreta spectrum, or PASS. The image to the right shows a case of PASS. The top part of the image showed a normal Plancita invasion, where uterus wall and Plancita are well separated, while the bottom part, you can see that Plancita, the dark part of the image, invade deeply into the uterus wall. Next. The overinvasion of Plancita into uterus makes it impossible for placenta to detach by itself, which it should. This leads to a life-threatening condition for the mother. Approximately 40% of patients with PAS needs blood transfusion. 80% of patients' uterus needs to be removed to avoid life-threatening bleeding. 10% of patients will suffer from damage to adjacent organs such as bladders. 2% of patients will face a risk of death. Next. The incidence of PAS has increased about 100 times since 1950s. The exponential increase of PAS is directly linked with the cesarean delivery rate, as shown in the graph to the, at the left bottom corner. If we track each individual patient, we also found exponential increase of PAS rate as times of cesarean section increases. Next. Cesarean section usually leaves a scar on the uterus. Clinically, it has been frequently seen that placenta evades along the uterine scar. However, it is unclear why and how uterine scar assists abnormally deep trophoblast invasion, which is a hallmarker of PAS. Next. To advance our mechanistic understanding of PAS, we developed an in vitro PAS model. Our model is inspired by the biophysical and biochemical properties of the native endometrial tissue. Using patient cells, we found that endometrium scar not only diminishes the resistance of endometrium to placenta invasion, but also actively assists placenta invasion, leading to PAS. Next. If you're interested, please join me in my poster session tomorrow. Thank you so much for your attention.
Hi, my name is Mohammad Mahdi Samandari. I'm a postdoc in Tamil Lab, Biomedical Engineering Department, UConn Health Center. And today I'm gonna present our recent achievement, which resulted in significant improvement in wound healing in large animal models. As you may know, different wounds are affecting a lot of people in the world, and current wound management strategies are not capable of performing or addressing the needs for those patients. And that's why we have millions of people suffering from overhealing or underhealing, which are like chronic wounds or like wounds with scarring, excessive scarring after the wound healing. So current development in tissue engineering and drug delivery resulted great promise. And one of the most promising approach in tissue engineering is bioprinting. Next, please. So in bioprinting, or better to say conventional bioprinting for regenerative medicine, the wound on the patient is scanned, and then a 3D CAD model is formed, and that CAD model is transformed to computer-aided manufacturing systems, which here is bioprinting, and a 3D scaffold is printed and then implanted into the defect. But this method is suffering from a lot of limitations. First of all, it needs the scanning modalities, CAD systems, like specialties for performing the CAD analysis and CAM bioprinting, which is not available everywhere. And second, it's delayed. So maybe by the time that the scaffold is ready and the wound is dynamic, you know that the scaffold is not fitting the wound anymore. So it's delayed and it's not like useful anymore. And finally, like the printed scaffold should conform completely into the defect and be fixed and integrate with the tissue, which is not happening because it's not completely fixed and it can move related to the tissue. So to address this, next please, we developed a simple approach, which is a handheld bioprinting strategy. So we developed a handheld bioprinter, which we call it biostylus. And using this method, the surgeon can inspect the defect and can print the scaffold, which may contain like cells or bioactive factors directly inside the defect, and cross-link it or gellify the scaffold at the same place, which we call it in situ cross-linking. And this in situ cross-linking can cause tissue adhesion because the fluid, which is the bioink, we call the first material of bioink, can penetrate inside the tissue, and then it can gellify or cross-link or solidify and maintain in place. So it can completely adhere to the tissue. So it doesn't need any fixation modalities, any CAD or CAM experience, or devices or scanning modalities. And it's based on like the surgeon decision, and it's completely based on the dynamic nature of the wound. Next, please. So using this method, we try to improve the wound healing in large animal models, which are pigs. And as you may know, pigs are a very similar species regarding the wound healing mechanism to humans. So we selected pigs, we created the wounds, and we printed the material in situ using our method or our handheld printer, and the material which is gel mount inside the defect, and the material completely adhered to the defect, and we were encapsulating a drug inside the material, which was releasing into the wound to increase the wound healing rate and improve the nature of the wound, which it resulted in a significant reduction in scarring and advanced wound healing in the porcine model. So I'm not going to mention this. I'm not going to mention more results here because definitely I don't have time. But if you are interested, you are welcome to join our presentation tomorrow at 3 p.m. Thank you.
Hi, this is Michael Arul. I'm going to talk about our recent project, injected nanocarriers for invertebrate disc repair and regeneration. Every year in US alone, approximately 6 million people suffer from IVD-related injuries, resulting in back pain and disability. Pharmacologic treatment to IVD include pain management, growth factors, or small molecule drug delivery to manage pain and disease progression. Injectable hydrogel are preferred to deliver the therapeutic agents within IVD. However, hydrogel carriers have several drawbacks, including quick dose dumping while acclimating in IVD itself with poor degradation. Injectable nanocarriers can overcome some of these challenges faced by the injectable hydrogels. It has a property of sustained release of therapeutic agents, quick degradation, while allowing repeated injection for continuous treatment. Here we report synthesis of fluorescent theranostic nanoparticles for tissue engineering applications. The main aim of this study is to deliver leucine-rich repeat protein phosphatase, that is PHLPP1, for age-related IVD degeneration. Fluorescent molecules were synthesized by using citric acid having high emission wavelength between 450 to 550 nanometer and conjugated with cellulose acetate polymer, a FDA-approved pharmaceutical excipients. The nanocarriers were characterized for their physicochemical properties, in vitro drug release, in vitro cytocompatibility, and in vivo injection in mice and imaging. Here, the figure 1 shows the schematic diagram for the synthesis of cellulose acetate conjugated fluorescent compound. The preparation of nanoparticle is carried out by using oil and emulsion technique, and same morphology showed that nanocarriers are spherical in shape. The particle size for the nanoparticles were found to be 280 to 300 nanometer. We studied the in vitro drug release using hydrophobic drug indomethacin and hydrophilic drug 4-aminopridine. The hydrophilic drug are freely water soluble, whereas hydrophilic drug is poorly water soluble, and it makes challenge in bioavailability of this drug during the drug release. The encapsulation efficiency of hydrophobic drug was found to be 80%, and for the hydrophilic drug, it was 40%. In vitro release, the hydrophilic drug showed sustained release of up to 7 days, whereas the hydrophobic drug showed up to 5 days. This confirms that nanoparticles can be used for continuous treatment and achieve the bioavailability. Figure 3 shows that in vitro cellular uptake nanoparticle using fibroblast cells, and it showed that nanoparticles are uptaken by cells. In vivo injection of nanocarrier into mice IVD was studied by using 25 gauge needle. The image revealed that nanoparticles were uptaken by NP cells. Further, it confirms that prepared nanoparticles are biocompatible in both in vitro and in vivo. In conclusion, the fluorescent nanoformulation developed in this study will allow platform for delivery of therapeutic agents for IVD regeneration. This nanocarriers allows repeated injection for continuous treatment of IVD regeneration. Further studies are ongoing to develop this technology for other tissue repair and regeneration. For more details of this project, please visit our poster session tomorrow. Thank you.
Good afternoon, everyone. My name is Shirley Koshi Chentateo. I'll be talking about the agent-based models employed by us to understand multi-species microbial biofilms. There's a lot to unpack in that sentence, so let's start. What are biofilms? Biofilms are formed when microbes attach to surfaces using extracellular polymeric substances. It is this structure that makes it hard for antimicrobial drugs to penetrate. Thus, biofilms are a source of infection on medical implants and in the oral mucosa. We look at the interaction of the pathogenic bacteria, Streptococcus aureus, and the probiotic Lactobacillus paracasei. S. aureus is known to promote the virulence of fungal biofilms, and L. paracasei acts as a deterrent. Our experimental colleagues grew the two bacteria as a biofilm to observe their interactions. On the top right, we can see 3D images from both the single and duo biofilm. The green depicts S. aureus and the red L. paracasei. As can be seen on the first image of the top right, when grown alone, S. aureus thrives. But when grown with L. paracasei, which is the third image on the top right, the growth is stunted. As I mentioned before, we use a technique called agent-based models to investigate this phenomenon. An agent-based model is a mathematical model with an environment containing some agents which follow some rules. In our model, the agents are S. aureus and L. paracasei. They follow the rules for growth, decay, death, and mechanical interactions. The model uses differential equations for the growth as well as for diffusion of nutrients within the biofilm. The bacteria move by shoving each other out of their personal space. The environment in our model involves a substratum for the bacteria to attach, the biofilm, and the liquid above the biofilm containing the nutrients. We've explored 4 different interactions, namely competition for nutrients in space, inhibition of growth of S. aureus by L. paracasei, production of surfactants by L. paracasei, which cause both the bacteria to detach from the biofilm and become planktonic, and the combination of inhibition and surfactant. The image on the bottom right shows the results from these 4 simulations. Again, green stands for S. aureus and red for L. paracasei. In the surfactant and inhibition plus surfactant images, that is the last 2 images on the bottom right, the floating spheres are the planktonic agents which detached from the biofilm due to the surfactant. As can be seen in the images on the top and bottom right, simple competition really does not explain the experimental observations. Since we observe in the competition model, the green S. aureus actually outgrew the red L. paracasei. Whereas in the other 3 models, we see that the growth of S. aureus in the biofilm is significantly lower than L. paracasei, with the inhibition plus surfactant model having the worst effect. My colleague, Dr. Linda Aschenbaugh, will talk more about the experimental side of the project later on in the program. If you want to learn more about these mathematical models, visit me at my poster in the group 3 room tomorrow. Thank you all for attending.
All right. Hi, guys. My name is Carissa Orlando, and I am in the psychiatry department. So I am looking at additional treatment factors that might impact anxiety treatment in school-aged youth. So Anxiety disorders are incredibly common in youth, and we have gold standard treatments that we know work for reducing symptoms of anxiety, but a lot of children don't get those treatments or can't access them. But at the same time, school nurses are often the first people that identify these youth with anxiety, because youth present to the school nurse with symptoms they think mean they're sick, like stomach aches, headaches, their heart pounding, things like that. So my team has developed an intervention that is based off of evidence-based treatment for anxious youth that can be delivered by school nurses. So this was piloted a couple years back. We compared CALM, that's our evidence-based intervention that's developed off what we know works, and we compared that to CALM Relax, which is an active control that just looks at teaching students relaxation skills. And so 21 nurses developed or implemented this intervention to 54 students. And we actually found that while both interventions significantly reduced symptoms of anxiety in children, there wasn't any superiority or any differences between the two interventions. So our evidence-based intervention performed pretty similarly to our active control. So what I looked at is what else could be going on here and what else could impact these outcomes in anxious youth. So does this have to do with something with the nurse? So the nurse's years of experience in working with students, their education. Does it have to do with how the treatment was delivered? So the nurse very rigidly adhering to the protocol, the nurse's use of soft skills in developing relationships with the child, how many sessions the child got, and then how engaged the child was in treatment. So what else can impact a decrease in symptoms? And what we found was the nurse's use of common structural elements in treatment that isn't necessarily related to any one specific treatment was significantly related to decreased symptoms. So the nurse working towards an identified goal with a student, the nurse teaching the student a skill and having the student practice the skill to help reduce their anxiety, the nurse using handouts and other materials to engage the student in their treatment, and then the nurse assigning out-of-session homework for the student to practice. All of these were significantly related to reduced symptoms. And this is all very good news because these are fairly easy things that nurses can incorporate into treatment regardless of what they're doing with the youth. And we know that it can help the youth experience fewer symptoms of anxiety and do a little bit better in school and elsewhere. And this also shows some good preliminary evidence that nurses can be professionals within the school that can identify students with anxiety and successfully treat these students to help them do a little bit better in school and in their lives. I would love to talk with you guys further about this. Feel free to stop by my poster tomorrow afternoon and learn a little bit more about this. Thanks so much, guys.
Hi, everyone. My name is Shir Ginsburg. I am in the Department of Public Health Sciences. We have been running a study on the problems that people were facing during the beginning of the COVID-19 pandemic. Back when there wasn't a lot of data on the disease, people were getting the information from a variety of sources. And there was a lot of confusion over who to listen to, and face masks versus hand sanitizer and things were out of stock. So there were a lot of uncertainties and challenges. And there was a major disruption of occupational, educational, and personal lives. And there was particularly a negative effect on mental health outcomes. And there was a blurring of the work-life balance, particularly for women who were working from home, but also managing a lot of the household duties that their partners were not managing. So there was also limited data on COVID, which was leading to widespread misinformation and ambiguity about how to protect against it. Different news sources were presenting different information because there's no clear federal guideline on what to do. We developed an anonymous survey at the beginning of the pandemic to assess participants' knowledge, attitudes, and behaviors around COVID. This was limited to Connecticut residents who are 21 and over, who don't work in healthcare. We were advertising the survey on social media and word of mouth. UConn Health, public health faculty and students distributed it around the end of March of last year. We got almost 500 survey responses to the survey. We analyzed it using SPSS, and we focused primarily on the types of problems that people faced during the 2-week shutdown at the beginning of the pandemic. Some of the issues that people faced in the pandemic at the beginning were anxiety, and women struggled during the pandemic more than their male counterparts, level of education, and news sources, and ambiguity about who to trust and who to believe. Problems associated with the pandemic, high levels of anxiety, higher numbers of sources of information. The more sources of information you turn to, anything from CNN to Fox News to newspapers to Facebook to the friend down the street to healthcare to clergy, the more sources of information, the more confusion. At lower age, people who are younger tended to report a higher disruption to their social lives and also higher disruption to their education. And it was harder for them to find jobs. And identifying as a woman, as I said, for women, it was harder to balance job obligations with home obligations and things that they were doing around the house, which were exacerbated because everyone was stuck at home. And so people were facing a lot of different things, unpaid household labor, sources of information, and the media was also emphasizing a lack of resources like toilet paper, which added to the mental distress. We would always see on the news about lack of toilet paper, lack of hand sanitizer, grocery stores being empty. So we're still in the process of analyzing some of the results, but we look forward to sharing that with everyone once it's available. So thank you for your time.
Hi everyone, this is Debolina from Evo Galactic Lab. Today I'll be talking, giving you a very brief, a simplified overview of the work we are doing here at lab. And we are trying to address the question if migrant therapy can lead to impaired fracture healing. I'll go over our neuropeptide which we work on, CGRP or calcitonin gene-related peptide next. So it's a 37 amino acid neuropeptide and it's a sensory nerve fiber. It's one of the most potent vasodilator. CGRP acts through its receptor, and which is made up of 3 receptor complex. So the first one is CLR next, which is calcitonin receptor-like receptor. Next is RAMP1, and the last one next is RCP, or the receptor component protein. CGRP is well known for its integral role in migraine-associated pain. Next. So, as you know, migraine pains can be really excruciating. And what it's known is next, that in migraine-associated pain, if you look at this picture here, compared to the resting CGRP levels when there is a migraine attack or when the migraine episodes start, the CGRP levels can reach up to 5-fold or even more higher. So it is logical that there are treatments which are actually targeted towards inhibiting CGRP or its receptors or blocking them. Next. So this is just an example of some of the FDA-approved drugs. So these are targeted towards antibody therapy, but there are also small molecule inhibitors or GPANs. There are also antagonists. So several anti-migraine therapies which are targeted by targeting CGRP blocking. However, on the right, you will see now I'll talk about bone fracture healing and CGRP's role. Next. So when a bone fractures or it's broken. Series of events takes place. I'm not highlighting on everything, but as you will see, a lot of neuropeptides and several neurotropic factors come to kind of, they are orchestrated together to help the bone heal. Next, I'm highlighting here on CGRP, which helps in bone healing by increasing the bone formation by osteoblastic activity. It also decreases bone resorption by decreasing osteoclastogenesis. So overall, it's helping in the bone healing. Next, so recent literature have already shown that CGRP regulates in bone metabolism. Next, and also in this recent paper last year, it showed that CGRP's role in bone healing. So if you compare your left and right, on your left you have this promising therapy for migraine, on your right you have this bone healing thing. Next, so this again comes back to our question whether migraine therapy can lead to bone fracture impairment. Next, so the short answer is yes, and that's what we are working in our lab. We are working both genetic models and pharmacological inhibition. Next, to know more about our work, please visit tomorrow at Group 5 poster. Next, and thank you very much for your time, and good luck to the rest of the participants.
Hi everyone, my name is Bingshen Wang and I'm from Center on Aging, UConn Health. My topic is P20 high senescence and aging. Aging is the biggest risk factor for most of our chronic conditions, and over the past 150 years, life expectancy increased dramatically, but not all of the gained years are healthy. And it was investigated that slowdown in aging that increased life expectancy for 1 year, it will be worth $38 trillion. And there are several hallmarks of aging, such as stem cell exhaustion, telomere attrition, and cellular senescence. Together with the disease-specific risk factors, it will cause diabetes and Alzheimer's disease and cancer. Such as senescent cells will be induced by DNA damage and oncogenic insult, and the senescent cell will stop proliferation. It will secrete a lot of pro-inflammatory cytokines, chemokines, and termed as senescence-associated secretory phenotype. P16 and P21 are the two major cellular markers for senescent cells. Now, most of the studies are focused on the P16 expression, and they found eliminate P16 senescent cells will improve physical function and in the aged mice, and also extends lifespan. But not all of the P16 positive cells are senescent cells, and also not all of the senescent cells are express P16. So we focus on another cellular markers, which termed as P21, and based on our previous studies, which shown in the top right, we found the P16 cells and the P20 cells are very different cell population from the flow cytometry data or the single cell data. Although they have some overlaps, they are different. So our lab generates a P20 model. It was driven by the P20 promoter, and it contains inducible Cree, and also followed by the GIP, and crossed with other Floxed mouse model, we can monitor and image and also eliminate these P20 high senescent cells and observe lifespan or health span in the aged mice. Also, we will examine the role of NMCAR pathway or other pro-inflammatory related pathway in P20 high cells in the age-related health span and lifespan. So I will be in the group 7. If you will learn more, welcome to my post. Thank you very much.
Good afternoon. This is Li Chao. I'm also a postdoc from Dr. Ming Xu's lab at UConn Health Center on Aging. So my study is about investigating the role of P21 high cells in insulin resistance. So insulin resistance refers to impaired response to insulin in tissues such as fat, liver, muscle, which will cause the pancreatic beta cell dysfunction and result in abnormal insulin secretion. So it has been found to increase the risk for many diseases such as type 2 diabetes and hypertension. So a number of factors has been reported to be involved into the development of insulin resistance, such as obesity and aging, where the senescent cells accumulate. So the cellular senescence is a stable and terminal state of growth arrest in which cells will stop proliferating. The senescent cells can secrete many factors, including cytokines and chemokines, which will promote the chronic inflammation and thus contribute to the development of insulin resistance and type 2 diabetes and its complications. So P60 and P21 are the two major cellular markers for senescent cells. To date, most senescent studies are using P16 transgenic mice model due to the limitations of the current mice model. Well, the efforts on P21 high senescent cells are imperative. So in view of this, our group generates a new P21 promoter driving Cray transgenic mouse model, by which we will monitor whether P21 high cells will accumulate with obesity. So by crossing this model with other flocks mouse models, we will kill or modulate the P21 high cells in vivo to see whether targeting these cells could alleviate metabolic dysfunction. Furthermore, we will collect obese mice and human fat with or without P21 high cells removal. By transplanting this fat into the mice recipients and testing their metabolic function, we will further demonstrate the role and underlying mechanisms of P21 high cells in the metabolic dysfunction. So our work will also provide a tool for further discovery of anti-aging medications. If you would like to hear more about our work, please join tomorrow's poster section in group 4 at 3 p.m. Thank you.
Good afternoon, everyone. My name is Momita Bhattacharjee, and I am from Lorenzen Lab, CCI. So today I'm going to talk about my research work, which is mainly focused on developing injectable amnion hydrogel as a delivery system for adipose-derived stem cells for osteoarthritis treatment. So osteoarthritis is a chronic, painful, degenerative joint disease which affects millions of people worldwide. The early symptoms involve inflammation of the synovial membrane. And as a result of this inflammation, pro-inflammatory cytokines are produced, which then activates the matrix degrading enzymes. And then these enzymes leads to chronic degradation of the articular cartilage. So the current treatment includes both non-surgical and surgical therapies, which provide temporary relief, but have failed to treat the disease pathogenesis. Thus, developing novel therapies which can suppress inflammation and regenerate and activate regenerative pathways can overcome the challenges associated with the current therapies. Recently, stem cell therapy using adipose-derived stem cells has gained significant attention, mainly because they can suppress inflammation by inhibiting the activity, growth, and proliferation of immune cells and can release various anti-inflammatory and counterproductive agents. However, there are also certain limitations associated with stem cell therapy, such as the localization and retention of these cells within the target site for a required period of time. And also there's a lack of optimum delivery systems. Thus, to overcome these challenges, we have developed a novel injectable amnion-based hydrogel, which can deliver the cells and localize them within the target site for a required period of time. Amnion membrane is the innermost layer of placental tissues, which are considered biological waste and are usually discarded after the birth of a baby. And the amnion membrane is considered to be highly biocompatible and contains various bioactive molecules such as collagen, fibronectin, laminin, proteoglycans, etc., and also has certain anti-inflammatory agents. Plus, with this, we hypothesize that if we can combine ADSCs with amnion, we can have synergistic anti-inflammatory and counterproductive effects to attenuate osteoarthritis. And so in our study, we actually isolated amnion membrane, decellularized it, and then solubilized it to form a solution. And when the solution is brought to physiological pH and temperature, it forms a gel. So at room temperature, it's a solution. And then when it is injected into the body at 37 Celsius, it forms a gel. So we then evaluated the biocompatibility of these hydrogels, and we evaluated their effect or their potential to support the cellular functionalities. And we found that the amnion-based hydrogels can support ADSC growth and proliferation. We then evaluated the potential of the amnion hydrogels in combination with adipose-derived stem cells to attenuate osteoarthritis in a rat osteoarthritic model. And we found that compared to the sham group, which shows a beautiful, smooth, intact cartilage, as shown by the prominent safranin O staining at the femoral and tibial ending, the combination of AM and ADSC also preserved the cartilage matrix with a strong and prominent safranin O staining. Thus, it shows that there was a synergistic anti-inflammatory and counterprotective effect from the amnion and ADSC group. Thus, to conclude, we can say that this novel hydrogel system has the potential to bring a major breakthrough in stem cell therapeutics and osteoarthritis treatment. Thank you. For more information, please join me for tomorrow's poster at 3 p.m. Thank you.
Good afternoon, everyone. Thank you for joining today. I'm Takayoshi Otsuka from Dr. Lorenzen's lab. Today's topic is bottom-up approaches for regeneration. My ultimate research goal is to achieve complex tissue regeneration, such as limb regeneration in mammals. My short-term goal is to identify the signals that enhance the regeneration ability. We can gain a lot of insights from the field of developmental biology, and by understanding how to develop or regenerate the tissues and organs, we can transfer these mechanisms to the mammalian system. Next. So here is the basic concept of tissue engineering. One of the goals of tissue engineering is to create a biomimetic environment by combining cells, scaffold, and signaling molecules. And tissue-engineered 3D scaffold can be used as a biological substitute of damaged tissue. This approach could successfully facilitate single tissue regeneration, as you can see here. However, we cannot achieve complex tissue regeneration, such as limbs. Next, please. So it is important to provide appropriate signaling molecules for the complex tissue regeneration because they can control the cells to differentiate towards a specific cell fate. So which molecules we can use for tissue engineering? Next. We focus on the fibroblast growth factor 8, FGF8, from the insights of developmental biology. FGF8 signaling has an important role in limb development and regeneration. The left images show the abnormal limb and digit morphogenesis in FGF8 deficient mice. The right image shows the reactivation of FGF8 expression during axolotl salamander limb regeneration. Interestingly, FGF8 expression is rarely found in adult mammals. So also the essential role of FGF8 was identified in early 2000. No one has utilized FGF8 for tissue engineering. Next. We investigate the effect of FGF8 on the proliferation and differentiation of mesenchymal stem cells as a model of limb mesenchyme. I won't explain the details here, but we found that the FGF8 supplement can control mesenchymal stem cell fate into various cell types. Although the single use of FGF8 may be difficult to restore complex tissue regeneration, but this fundamental knowledge can be used for the tissue repair. Next. So now we are working on the use of the FGF8 as a new therapeutic to enhance regeneration. In conclusion, a deeper understanding of the development or regenerative process has the potential to inform us to design novel and translational strategies that can enhance the regenerative abilities in humans. So if you are interested in this work, please join the poster session. My poster is in group 4, 3 to 4 p.m. tomorrow. That's all of my talks, and thank you for your attention.
All right, good afternoon. I'm Stefan Somsky. I work in the Department of Neurology under Dr. John Greenfield. And I'll be talking today a little bit about seizure termination and how we figure things out about that. Next, please. So generally speaking, you are all familiar with epilepsy and seizures. Statistically, we all know someone who has it. And after years and years of research, we're getting close to understanding many different components of it. For example, the way that seizure starts, proceeds, and in theory, terminates. However, next, please. We don't really know how a seizure terminates. We know it runs off. There's a couple different theories. In some cases, we suspect that the rest of the brain shuts down the signal. In other cases, we think it wears itself out. And the way that we look at that is very difficult, partly because of how much noise, physical movement, artifact, and everything else you see in MRI and EEG, which is the way that we look at this problem. Next, please. We think that because we've had a great deal of success, both personally and as a field, using network analysis to see how seizure states change, how seizure approaches, and so on, and even investigate how well various treatments work on it, this could work for seizure termination as a question. But there's a problem. Next, please. Generally speaking, people form networks using correlation, synchrony, correlation between various phases and everything else, but that doesn't always work in seizure. Next, please. Where because of the heavy level of synchronization in that EEG signal, most correlations that we see are suspect. You could politely call them spurious or impolitely call them noise. So we need a way that gets away from correlation, next please, and into a method focused on causation, what is actually happening in the brain, not just correlated connectivity, but what's called functional or effective connectivity. And next, please. Our method for doing this is a multiple input, single output, or MESO, state-spaced, model-based network estimation. Next, please. What we do is take recording from a huge portion of the brain using intracranial EEG, treat every electrode in that point as the source of a system, and estimate how much everything else in that system affects it by creating a model that accurately predicts the behavior in that node, and then reverse engineering that model to see which parameters and thus which channels are most critical here. Next, please. So for instance, in this case, we look at that B term, which is our input variables, our input parameters. And if they're above a certain significance, we can find that there is a meaningful connection there. Once we have this, we can repeat the process over the entire network for various periods of time during seizure and watch when changes in that network occurs as termination approaches. Next, please. And what we see ultimately is two specific things. One, that the seizure onset zone, that core part the seizure comes from, begins to wear itself out. And also simultaneously, there's a rebound inhibition from surrounding tissue, meaning that the two arguing sides of the field are both half right. And the exact way in which they get to that point, we're investigating more closely now using this reverse engineered model process. And this has longer term implications for everything from drug design, assessment of how seizures end, differentiating various clinical states, and ultimately, once we know how seizure ends naturally, things like neuromodulation will become more effective at creating that terminating state artificially, letting us proceed further towards a seizure solution. Thank you for listening, and I'll be at group 5 tomorrow from 4 to 5 o'clock. Thank you.
Sorry, I was Muted. My name is Alisa Burkhardt, and I'm going to talk about how we measure phantom or how to test tinnitus. I'm a postdoc in the Oliver Lab, which is a lab in the neuroscience department. So first of all, why do we care about tinnitus? Next, please. 25 to 50 million just in America report that they have had some experience of tinnitus in the last year. And just to give you an example how annoying that could be, next, please. So if you imagine you have that kind of a sound going on the whole time, 24 hours a day, 7 days a week, when you try to fall asleep at night, if you have to study for an exam, it will not shut off. So next, please. You can imagine why a third of those people reported to be a moderate to very big problem in their life. So another problem with the tinnitus patients is next. Currently, there's no cure. The only therapy available, which kind of helps a little bit, is to learn to live with tinnitus, but there's no pill you can take which will make it disappear. And therefore, there's a desperate need to find a cure, so to say. So next, we need an animal model to test our treatment options. However, there we have the next problem. Next, how do you ask a mouse if it has tinnitus or if it got better after your treatment? So one approach is next to use a behavioral test. For example, in this case, you teach the mouse that silence is safe, but if there's a sound, and it can move around freely, but if there's a sound next, it has to go to the other part of this test box. So it learns that if it's silence, if there's silence, it's safe. If there's a warning sound, it has to move. However, if it has tinnitus, this tinnitus sound is safe. So there's no penalty for staying there if there's a tinnitus sound. So if the warning sound is very similar to the tinnitus, it might not perceive it as a warning sound. One big problem with those behavioral tests next is that those behavioral tests take a very long time, many weeks, and still only let us infer if tinnitus is present. Additionally, the field has many different behavioral tests available and is not always in agreement which test would be the best. However, next, the one thing everyone is in agreement is that tinnitus is a brain problem and not an ear problem. So the hypothesis of our lab is that not only in silence something must be wrong or different in tinnitus brains, but next, actually sound processing is different in tinnitus versus non-tinnitus brains. So our approach is to directly record from the source. So next, we record neuronal activity in the auditory midbrain in mice, and we test the response to several different acoustic stimuli and compare tinnitus versus non-tinnitus mice or brains. And additionally, next, we also use a non-invasive recording technique called auditory brainstem response, which is the same as what you use in neonatal hearing screens. And we do that in mice and humans, both with tinnitus and without tinnitus, and try to find a difference in the response pattern. And if you have been intrigued enough by me to learn a little bit about more what we found or how we give mice tinnitus in the first place, please next come and visit Chris Lee, my colleague, who is going to present a poster about our experiments in group 5 tomorrow between 4 and 5 p.m. Thank you.
Hello. Hi, my name is Neeraj Singh. I'm a postdoc in the Department of Neuroscience working with Dr. Yicheng Yan, and we work on Alzheimer's disease. So the topic of my talk focuses on exploring the role of microglial base 1 in Alzheimer's disease. Now, Alzheimer's is a neurodegenerative. Next, please, just do 4 next. Yeah. So Alzheimer's is a neurodegenerative disease, mostly affecting the elderly patients, and there's a chronic, there's eventually loss of memory. And if you look at the pathophysiology, a lot of things are done, but nothing substantial is coming, except for the fact that there is always an initial event, which is the deposition of amyloid plaques. And these plaques are made up of amyloid peptides, which are the neuronal APP processing product. And this is being mediated by beta secretases followed by gamma secretase. So this beta secretase is base 1. And what's done that since base 1 is a rate limiting step, so therapies revolved around targeting this base 1 in knocking it down so that patient will be, there won't be much amyloid deposition and there won't be loss of neurons. But all the therapies failed because of either toxicity. And the other thing is that base 1 is required for neuronal nervous system development. So what we did was we made a mouse in which conditional knockout mouse that we pass the developmental age, and at 2 months we delete this base 1. So in this figure on the lower side, you see that there is a plaque deposition till the middle panel is 75 day. It's both top is the control, the bottom is the base knockout deletion. And you see at 300 days, like there is literally no amyloid plaque, which is with this red color here in the base knockout, and there was improvement in memory. So what we took from there is that, okay, neurons stop producing amyloid peptide because base is inhibited. And second thing is whatever peptide was present, it was being cleared out. So we thought maybe the neuronal base 1 also have a role in phagocytosis. Next, please. So we did a single cell RNA-seq on these mouse brain, and we find out that indeed there is a signature change in these microglial cell population. There are 3 types of microglia based on subset of gene. There's homeostatic DAM1, DAM2s. So DAM2s are neurodegenerative type microglia. And what we see in this third panel, if you knock down base 1, these neurodegenerative type microglia has gone down. So that was something very exciting. Next, please. Next. So what we saw, first we confirmed that whether base is expressed in microglia. We did single cell RNA-seq in control and 5x FAD, it's an AD mouse model, and we saw there is increase in base 1 expression. We confirmed with the Western blot in vitro treatment. And what we find was there was an increase in phagocytosis in these base 1 knockout microglia. And same thing was associated with base inhibitor. If we treat these microglia with base inhibitor, there was increased phagocytosis. Next, please. Next. So then what we did, we looked at base knockout mice, isolated microglia, and did single cell RNA sequencing. And we see that there was a very distinct primed population of microglia in base 1 knockout, which means that they are primed for uptaking amyloid challenge. And then we did systematic intracellular signaling, looking at PI3K-RAC1 signaling. Next, please. Then we looked at glycolytic pathway. It was enhanced in these base knockouts. Next, please. Next, please. Next. Then what we did is we did a conditional base 1 knockout in microglia specific and did the single cell RNA-seq in 5x FAD mouse model. And what we saw was that there was significant reduction in DAM-associated microglia in base knockouts. And this was associated with a specific subset of genes, which I'll talk tomorrow. And what we see, there was a 25% reduction in plaque production. Next, please. And there was a reduction in LTP was restored, so that was there was a preserved the synaptic plasticity was preserved in 5x FAD model. So we present base 1 microglial base 1 as an alternate therapy for Alzheimer's treatment. Thank you.
Hello, my name is Kevin Child. I'm a postdoc in Dr. Cotney's lab. Today I'm going to talk to you about a splicing program for heart development. Next, please. So starting with the problem, congenital heart defects affect approximately 1% of live births worldwide. CHDs range from mild to severe, requiring surgical intervention within the first year of life. There are several known causes for CHD, from environmental hazards, de novo mutations in familiar forms of CHD, which account for less than 10% of all cases. Chromosomal abnormalities contribute to 20% of CHDs. and there is strong evidence for a genetic component for CHDs. However, approximately 60% of these cases still remain unexplained. Recent evidence has pointed towards the importance of alternative splicing during heart development, which can help guide our efforts for investigating the unknown causes of CHD. Next, please. The Caseman Lab Group has recently published data profiling multiple species and tissues during development, looking specifically at alternative splicing events. They found several alternative splicing events across development in all tissues, and they found that 2 specifically segregate from all the other tissues. We see the heart and the brain are segregating. And this is something that's conserved across species. And we see that there's also a developmental trajectory in these splicing events. Next, please. Our lab recently profiled the epigenomic and transcriptomic changes that occur during the earliest points of heart development, starting from CS13 to Carnegie stage 23, where we looked at several novel regulatory regions as well as co-expression network analysis. This revealed a hub of genes that is part of RNA processing and splicing, which is dynamically differentially expressed during this period. Next, please. So to investigate this period of development, I use several splicing tools that the field has developed to look at specific events across these Carnegie stages. I also use annotation-free alignment to investigate novel splicing events that may occur during this early period of development. And next, please. Next, oh yeah. And then to validate any of these findings, our lab has recently acquired nanopore minion, which I use for long read sequencing in order to get full transcripts in order to see these splicing events. Thank you very much. Hope to see you at the posters.
Hello everyone, I'm Feyza Aymaz and I'm a postdoc at the Jackson Laboratory in Lee Lab. And today I'm going to talk about one of the most complex regions of the human genome, 3q29, which is also associated with genomic disorders. So genomic disorders usually occur as a result of chromosomal rearrangements. Next, please. These rearrangements usually caused by small duplicated regions, which you can see in yellow arrows. And depending on the orientation of these duplicated small blocks, we observe different types of structural variants. In the first example, we're looking at duplicated blocks which are in the same orientation and results in deletions and duplications. Next, please. And in the next one, we're looking at inverted duplication blocks, which results in inversions. Next, please. And in the last one, we're looking at combination of both duplication blocks in the same orientation, also inverted orientation, which results in complex structural variants. So some of these structural variants cause genomic disorders. And one example is 3q29 deletion syndrome. Next, please. So in this image, we're looking at probands with 3q29 deletion syndromes. One of the common phenotypes observed in these patients includes facial abnormalities, cardiac defects, and interestingly, this deletion syndrome is associated with a greater than 40-fold increased risk for schizophrenia. So it is important to understand the underlying structures causing this disorder. However, next, please. This is, like I said at the beginning, one of the complex regions of the human genome because of the small duplicated blocks. So this is a representative of 3Q29 region, and arrows represent the orientation of small duplications in this region, and shaded areas show the duplicated blocks. So this is basically combination of duplicated blocks in direct orientation and inverted orientation, which causes complex structural variants. Next, please. So my research question was to identify or to understand if there are certain haplotypes or certain structures in this region which may cause 3q29 deletion syndrome in individuals. Next, please. So to answer this question, I use optical mapping data to characterize 3q29 region in individuals with 3q29 deletion syndrome, their parents, and also unaffected individuals. And once I identified haplotype structures in our sample cohort, I validated or confirmed haplotype structures by using long-read sequencing data. Next, please. And our results show that there are 37 distinct haplotypes in this region. And all these colored arrows in the last image represent different types of structural variants observed in each haplotype. So if you want to hear more about our results, please attend our poster session tomorrow at 4 p.m. Thank you.
Hello, everybody. My name is Mikhail Dogan. I'm a postdoc at JAX. Hope your day is going well in this postdoc appreciation week. By the way, postdocs, we appreciate you. Thanks, guys. Keep up the good work. This was a COVID-19 antibody study published in the Nature group back in January this year. I'm going to start from the box number 1 and go with the box numbers from there. During the pandemics, we worked really hard and developed a bead-based antibody assay to measure the antibodies of COVID-19 patients with flow cytometry. That assay worked quite well, and we wanted to compare it with the other stuff out there. So we went to Walmart and bought an ELISA COVID-19 assay. Actually, they were selling it in GeneScript for 11 shekels. That was too much money. Anyway, as you can see in the box number 2, we correlated the results of the both assays by measuring the same plasma samples, and they confirmed each other, so it's good. And on the right, as you can see, we also compared the results of the both assays by measuring the same test results, and you can appreciate the dynamic range of our antibody assays. Skipping to box number 3, we were able to measure all sorts of antibodies. And when you combine just the 2 of them, you were able to get to 100% of sensitivity and specificity with our diagnostic test. So that's a good diagnostic test, I would say. And on the number 4 box, we made a fake SARS-CoV-2 virus because it's fun to make synthetic viruses and it's safer to work with. With that virus, on the number 5 box, we developed a neutralization assay to measure the neutralization capacity of the COVID-19 patients. And it turned out that the neutralization capacity of the COVID-19 patients correlated with the severity of the disease. That's interesting. Perhaps maybe their immune system fought really hard to win this war against the COVID-19. We also had some plasma donors. Those were some nice, good people who got COVID-19 before and healed from it and wanted to help COVID-19 patients by donating some plasma. And we were able to measure the neutralization titers, neutralization capacity of those patients, of those plasma donors. And it turned out that their neutralization titers varied a lot. Like, as you can see in the bottom, that one guy, they had antibodies, but his antibodies didn't work repeatedly. If you get COVID-19, you don't want to get the plasma from that patient. You want to get the antibodies from the top guy, which is like 50 times better than the average neutralization titers among the plasma donors. So that's really important, because if you can use the top guy's antibodies, you can heal 50 patients with the same amount of plasma. So that's really important to have a wide dynamic range antibody assay, I would say. On the number 6 box, we also wanted to compare the antibody levels of the vaccinated people and COVID-19 people. And it turned out that the vaccinated people had like 35 times better antibody levels compared to the COVID-19 people. So get vaccinated, I guess, and come on down to our virtual poster presentation tomorrow at 4 p.m. One presentation is only for 3 shekels, 2 for 5 shekels. It's almost free. We're also giving away some free enchiladas. Don't forget to bring your plumbus. Otherwise, how else you're going to get your free enchiladas? I mean, you have only 2 pockets. If you're confused enough with all of this, come on down to our poster presentation tomorrow at 4 p.m. Thank you for your attention. Bye.
Hi, everyone. I'm Domo Yang. I'm Domo Yang from Dr. Peng Wang lab. Today, my topic is UC ligase UBR5 positive regulates antibody innate immunity. Next, please. Generally speaking, the host have antibody innate immune system, including mainly 2 pathways. First is the SIG-STIM pathways, which censoring DNA virus infection, and the other one is RLR signaling pathways, which include RIGA and MDF5, mainly censoring RNA virus infection. Next. The ubiquitin system is is a widespread system to regulate post-translational protein modification, which include like E1, E2, E3 enzymes. In this, E3 ligase has been demonstrated involved in protein degradation and complex assembly and even deubiquitination, and also antiviral immune response as well. So we next. So we want to know if there are any other new E3 ligase involved in antiviral immune response. Next, please. So then we use a CRISPR-Cas9 system to try to screening some specific E3 ligase involved in antiviral immune response. First, we generate 375 individual lentivirus for CRISPR knockout in HEK cells. Next, please. And then we do transduction to FTGH E0 luciferase report cell lines. Next. And the next. And after 6 days purulizing selection. Next, please. We got the individual E3 ligase CRISPR knockout library. Next, please. And then we use different stimuli like ISD to analog dsDNA virus, and the poly-IC to analog RNA viruses, and also interferon beta to test the interferon signaling. And then we do identify a very specific one named UBR5 involved in the RNA virus anti-RNA virus signaling. Next. So now the next step is we're going to do in vitro study, use different viruses and also PPR ligand to know how does UBR5 regulate PPR signaling, and also in the vivo we now got the conditional knockout mice for this UBR5 genes, and to see if what is the physiological function of UBR5 during EMCV or SARS-CoV-2 virus infection in vivo. Thank you.
Good afternoon, everyone. I'm Tingting Geng from Dr. Peng Wang's lab. My topic is the Uvaccine 3B Control B lymphopoiesis well blink. Next, please. So The hematopoiesis is finely regulated to enable timely production of the right number and the type of the mature immune cell to maintain tissue homeostasis. Next. We found that Uvexin 3b play an important role in maintaining this process. We found the 3b knockout mice have significantly fewer B cells in blood, spleen, and bone marrow. It's about 10-fold lower compared to the 3b wild-type litamate. Next, please. Why this happened? We took a deep look at the B cell development. B cell development have several stages inside of bone marrow, including pre-progenitor B cell, progenitor B cell, large precursor B cell, small precursor B cell, and immature B cell. Next, please. By flow cytometry, we found that the large precursor B, small precursor B, and the immature precursor B was significantly lower in 3b knockout mice, while the progenitor B cell stayed the same. So this is suggesting that the Uvexin 3b is essential for transition into large precursor B cell. This transition process is controlled by a set of the cell intrinsic transcription factor inside the B cell, and also the cell extrinsic factors such like chemokine, cytokine, and growth factor in the bone marrow niche. Next. So to determine whether Uvexin 3b contribute to the cell intrinsic or cell extrinsic factor, we designed this two-way bone marrow transplantation. Next. We found that the transfer of the wild-type bone marrow to the 3b knockout mice will correct the B cell deficiency, but the reverse transcription transplantation couldn't. So the Uvexin seems to control early B cell development in a cell intrinsic manner. Next. Then, so the single cell RNA sequence, we found that the pre-B cell receptor, which also called the pre-BCR signaling, was impaired. Next. The activation of the pre-BCR leads to a assembly of a membrane-associated protein complex containing B linker protein, BLINK, SIG, BTK, PLC gamma 2. And we found that 3B may regulate pre-BCR signaling by targeting those components. So after examining protein level of those components, next, we found Uvexin 3b, the BLINK protein level was dramatically downregulated in Uvexin 3b knockout mice. This is especially in the early stage of the B cell. But the BLINK mRNA expression level remained the same. So this bring up our hypothesis. Uvexin 3b is essential for precursor B transition to large precursor B by regulating pre-BCR signaling and BLINK stability. Next. Without functional pre-BCR signaling and BLINK, both proliferation and differentiation of a large precursor B cell was impaired. Next. This dysregulated immune system will lead to 3B knockout mice highly vulnerable to viral infection like SARS-CoV-2. So this results showing that 3B is essential for maintaining homeostasis and particular for the B cell during the state and the viral infection. So if you want to find more detail of my project, please come to my poster tomorrow at group 6. Thank you.
Hi, can you hear me? Can you hear me? I cannot see my slide. Yeah. Okay. Hi, I'm Gautam, and I'm a postdoc at Department of Molecular Biology and Biophysics, and working with Professor Olga Peti. Today I'm going to speak about the regulation of the protein phosphatase 1 by inhibitor 3 molecule. As the name suggests, inhibitor 3 is inhibiting the protein phosphatase 1. So before going to the detail of the work, first I would like to give you a why and what we are doing. Basically, majority of the biological regulation is governed by phosphorylation and dephosphorylation event, and kinases phosphorylate the substrate, while phosphatase reverse the effect of the kinases. And these two proteins are not only acting as an enzyme, but it also regulates with several other regulators which interact and regulate several other biological functions like apoptosis, cell cycle progression, and transcription. So, but one of the issue in this entire system is in the past, people have paid a lot of attention onto the kinases group of family and identify more than 10,000 substrates. If you compare with the phosphatase, where only 400 regulators and substrates has been identified. So our lab professor Olga Empathy has spent around 2 decades time to understand the biological regulations and at the molecular level of this phosphatase group of family. So one of the class is serine threonine protein phosphatase, which accounts around 98% of the dephosphorylation event inside the cell. So it interacts with substrate and a regulator via short linear motif, which include RBXF5 arginine, silk motif, and active site. And here is one of the picture in section 1. You can see the different binding pocket onto the surface of the PP1. And in the section 2, you can see there are these are few of the crystal structure of with different regulators in complex with the PP1 has been solved from our lab. So you can see it recognize the different regulator recognize the different binding pocket onto the surface of the PP1. And as I said, we are working on the inhibitor 3 molecule. Inhibitor 3 is an intrinsically disordered protein, which is 127 amino acid lengthy protein, and it has the canonical RBXF motif. It is conservative in majority of the regulation. It interact this particular motif interact with the PP1. So we started our investigation using the NMR spectroscopy in section 4. At the section 4 on the left panel, you can see in the NMR spectra of the free, which is in red in color, and the black, which is in complex with the PP1. And you can see majority of the peaks are missing. It means that the I3 is interacting with the PP1. And the similar kind of experiment we acquired where we have abolished the RBXF, where we mutated the RBXF motif to abolish all the interaction of the RBXF motif with the PP1, and majority of the peaks are reverted back, leaving behind some of the peaks which is intact. When we critically analyze these two experiments, then we found that there are at least 3 binding motifs which is interacting with the PP1, and that is falling between 27 to 68 region. So we design a new construct, 27 to 68, and in order to prove that whether this particular 27 to 68 region manifest all the biological property and binding property, we did a combination of experiment which is SPR and inhibitory assay, and proven that this particular motif has all the manifest all the binding property as well as inhibitory property. So that particular construct has been taken to investigate for the co-crystallization study, and we have solved the crystal structure at 2 angstrom resolution, and we identified the 3 binding pocket onto the surface of the PP1 by this RVX and this I3 molecule, which is silk motif, RVXF, and CCC. And this particular result also complementing our NMR result. And further, we mutated the CCC motif. This is the new motif, new finding of the CCC motif of the I3, which we mutated to SSS motif, and we abolished the interaction, which is similar to the 27 to 59 region, and it also lose the property of the inhibition. It means that CCC motif in conjunction with RBXF motif is required for the inhibition of the PP1 molecule. So I would like to thank my lab member and and Professor Chris Heinen for organizing this meeting, where I got an opportunity to introduce myself. And thank you very much for for your kind attention.
Hello. Hi. Good afternoon, everyone. I'm Yuan. I'm very glad to present my research today. My title is HGF is a key determinant of a fibrotic kidney local microenvironment. I think the concept of the tumor microenvironment is familiar to us. It plays important role in the cancer progression and metastasis. But unlike the microenvironment in tumor, so far we little know about the concept of a fibrotic kidney local microenvironment in the kidney disease. The kidney local microenvironment is composed of kidney cells in the tuber interstitial compartment, including tuber cells, fibroblasts, parasite, endothelial cells, and inflammatory cells. In order to explore the composition and the regulation of a fibrotic kidney local microenvironment, we did some experiments. Firstly, we did RHC staining for kidney biopsy samples. These samples are from chronic kidney disease patients. We found that the fibrotic lesions were not homogeneous throughout the kidney. And we found that the tuber epithelial cells, macrophage, and the HGF-CMET pathway are potentially involved in building the fibrotic kidney local microenvironment. Secondly, we analyzed the single-cell RNA sequence data. We found that the tuber epithelial cells and the macrophage are the two major cell populations in a fibrotic kidney. Later, we created a mathematical model. It predicted that the loss of CMET in tuber cells would cause greater response to injury than in macrophage. Based on above data, we found that the HGF-CMET signal in both tuber epithelial cells and the macrophage will play the important role in regulating the fibrotic kidney local microenvironment formation. Later, we then generated the conditional knockout mice to delete CMET receptor in the tuber or macrophage. Then we subjected the knockout and the wild-type mice to the renal fibrosis model to induce renal fibrosis, including the ischemic reperfusion injury, the folic acid administration, and the unilateral urethral obstruction. We found that the specific deletion of CMET in the tuber cells will influence the epithelial plasticity and accelerate the synthesis of the extracellular matrix in the fibrotic kidney local microenvironment. But the loss of CMET in the macrophage will increase the risk of fibrotic kidney local microenvironment formation, but does not affect the speed of extracellular matrix synthesis. Thank you very much for your attention. Welcome to visit my poster tomorrow. Thank you.
Hello, everyone. My name is Roberto from Dr. Dongari's lab, and I will talk about probiotic bacteria and candida. I'm sure most of you are familiar with probiotics and with candida infections. Candida albicans is a member of the health-associated microbiota, and it's the first cause of death due to fungal infections. Lactobacillus johnsonii colonizes the same sites as candida, but there is not known what happened when they interact. Lactobacillus in general are known because of their probiotic effect on humans and other organisms, and some of them display antimicrobial activity, but johnsonii has not been studied for that. So we isolated Lactobacillus johnsonii strain, we sequenced its genome and evaluated its activity. If we see on figure 1, this is the Candida albicans versus Lactobacillus in different media on MRS broth, on BHI broth. And Candida is inhibited in the presence of the Lactobacillus. These two culture media are different, and when Lactobacillus is grown alone or with Candida, the media is acidified. On MRS, it's acidified to almost a pH of 4, and on BHI, it's like around 5.5. but regardless of the acidification, Candida is inhibited on similar ways. So we culture them on the biofilms condition, biofilm formation conditions, and we observed that also Lactobacillus johnsonia inhibited Candida growth, and the Candida biofilm was reduced, and the pH was also acidified. So in another experiments, the data are not shown. We saw that if we acidify MRS to pH 4 and BHI to pH 5.5, which are the final pH of the initial experiments, Candida on MRS doesn't grow, but on BHI, it does grow as a control. The acidification doesn't have any effect. So if we see the figure number 3, we explore the lactic acid production and the acidification of different culture media with Candida and Lactobacillus, and we see that the production of lactic acid is almost the same, the total lactic acid, but on MRS, the D-lactic acid in gray, it's a favor, but on BHI and RBF, the L-lactic acid production is favor. So we evaluated the effect of this acidification, and it seems that regardless of the acidification or the type of lactic acid production, Candida is inhibited. So it seems that the lactic acid or lactate and the acidification by itself alone, it's not the main factor to inhibit Candida. This is only a small part of the whole story. This is only like an introduction of how we are trying to understand the anti-candidal effect and interaction between these two organisms. They have probiotic potential for human health. So if you are interested in to learn more, you can see my poster presentation tomorrow at 3 p.m. Thank you.
Hello, my name is Linda Archambault, and I'm also a postdoc in Anna Dongari Bagtiglo's lab, and we're in the Department of Oral Health and Diagnostic Sciences. Today I'm going to talk about our work on multi-species oral biofilms, and you already heard a little bit about that from Shirley Koshi-Chentitail, and I'm going to continue that. So I wanted to tell you a little bit about the background and the reasons for this study. Our mucosal surfaces support many microorganisms, and most of the time our microbiota keeps us pretty healthy. Next, please. In patients with compromised immune systems, though, and other conditions, an imbalance in our microbiota often happens, and the normally harmless commensal microbes can become pathogenic. Next, please. Often, one or a few species grows uncontrolled, like in this poor mouth. It has an overgrowth of the yeast Candida albicans, also known as thrush. And here, Candida has formed biofilms on the tongue. Biofilms are organized microbial communities that adhere to all sorts of surfaces. Next, please. Microbes adhere, form a colony, and then continue to proliferate, encasing themselves in a matrix of extracellular polymeric substances called EPS. Biofilms protect microbes from our immune cells and from antimicrobial drugs, and this makes them very hard to eradicate, both on medical devices such as catheters and on our mucosal surfaces like in our mouth. Next, please. In the Dongari lab, we're interested in how the yeast, Candida albicans, interacts with bacteria in the mouth. Researchers in our lab had previously discovered that Candida, which is magenta in this image, and Streptococcus oralis, the bacterium in green here, synergize. So they make their combined biofilm more damaging than biofilms of Candida alone. Another bacterium, which Roberto just talked about, Lactobacillus, has an inhibitory effect on other microbes, and for this reason, Lactobacillus species are often used as probiotics. So we hypothesized that lactobacilli might interfere with the synergy between Candida and Strep aurelis and protect patients from damaging biofilms. So to explore the complex interactions in the biofilms, we decided to approach the problem from two sides. We use fluorescence imaging and other lab techniques in our lab to view and measure live biofilms. And our collaborators are mathematical modelers who grow and observe biofilms in silico. Next, please. They use agent-based models which treat each organism as an individual agent. Agent-based models are powerful because they can reveal emergent properties of complex systems like biofilms. By using a back and forth process between experimentation and modeling, we have been better able to make sense of these interspecies interactions. Next, please. In the first stage of our collaboration, we modeled interactions between Streptococcus aurelis and Lactobacillus paracasei. We found that L. paracasei has an inhibitory effect on S. aurelis growth in dual species biofilms. This bodes well for its potential to interfere with Candida S. aurelis synergy and thereby protect patients from overgrowth of Candida. If you are interested, please join me in the poster session tomorrow to learn more about our work on these microbial interactions in biofilms. My colleague Shirley Koshi-Chenthetale will present a poster on the agent-based modeling side of this project, and we are both in poster group 3 at 3 p.m. on Wednesday afternoon. Thank you very much for your kind attention.
Good afternoon, everyone. I'm Inhi, a postdoc at Jackson Laboratory. So today I'm going to talk about extrachromosomal DNA in cancer. So why is cancer so hard to treat? If tumor mass consists of identical cells, then the cure strategy for these types of tumor wouldn't be complicated. However, in most tumor, there are substantial differences among the cells in abundance and types of genetic materials. So we call these differences tumor heterogeneity. And it makes treatment strategy more complicated because treatment failure, development of resistance, and ultimately cancer recurrence. So understanding the tumor heterogeneity mechanism is very important to manage complex tumor more effectively. Next, please. In normal cell, we have a bunch of genes in chromosome, and the centromere on the chromosome is essential because this centromere is required to inherit the mother cell's genetic material equally to daughter cells. Next, please. However, in cancer, the major oncogenes were found on the circular DNA outside of chromosome, and we call this extrachromosomal DNA, ecDNA. And these oncogenic ecDNA are found in 50% of human cancer, and its clinical relevance with poor patient outcome was recently uncovered. Interestingly, ecDNA doesn't have centromere, thus it has been assumed to be unevenly inherited. However, there was no appropriate tool to explore its behavior in living cancer cells. Next. So my project was focused on developing an ecDNA tracking tool, which can tag multiple fluorescent molecules on ecDNA specifically. So we can see ecDNA molecules in living cancer cells under the microscope. Then I applied this tool to explore its behavior, and I observed mainly two things. Next, please. First, I observed the ecDNA segregation during mitosis, and as expected, ecDNA, each daughter cells gets a different number of ecDNA molecules at the end. And it is very important because this uneven segregation will generate substantial heterogeneity within only a few cell cycles. Next, please. Here are the schematic cancer cell division based on ecDNA uneven segregation model. At fourth generation, all cells have the same number of chromosomes. However, the amount of ecDNA in each cells are extremely various. So this first observation indicates that ecDNA can repeatedly generate tumor heterogeneity within only a few cell division. Next, please. Second, I observed the ecDNA generating its cluster. So two ecDNA molecules at the beginning come close to each other, and form clusters. And this phenomenon was observed in more than half of cancer cells. Next, please. And we found RNA polymerase II recruited into ecDNA clusters, and the cells with ecDNA cluster expressed more functional RNA. So these results show that ecDNA clustering serves as a transcriptional hotspot. Next, please. So in summary, ecDNA enhances substantial tumor heterogeneity through its uneven segregation and its cluster. So based on this method, I hope we can explore more unknown ecDNA biology, and we can re-engineer it for diagnostic or therapeutic applications. So please join me at a poster session tomorrow at 4 p.m. for more details. Thank you.
Good afternoon, everybody. So I'm a postdoc in the VIR-HAC lab at JAX, and I'll be going over my research on how epigenetic disorder influences cellular programs and drives heterogeneity in glioma. So just in general background, the initiation and progression of cancer is often marked by the accumulation of, yes, that's fine, of the accumulation. So I think there's some, can you go back a bit? Thank you. Yeah, so you can, yes, thank you. So the initiation and progression of cancer is often marked by the accumulation of mutations that alter the growth and survival of tumor cells. So these mutations are acquired in different proportions, and these different proportions or groups of tumor cells that can be referred to as tumor clones, they compete. Oh, okay. Oh, sorry. Okay, yeah. So something's happening with this. So I guess we can just leave that initial thing. But anywho, so yeah. So I guess leaving it right there is fine. So anywho, so this schematic sort of shows how. Okay. Well, I guess we can skip the first. Is this automatically progressing? Okay. Yeah. All right, so quick flash. And so this is a schematic of different tumor cell clusters. And this idea is, if you can see it quickly, you can ignore the details and sort of just look at the main picture here. But this is looking at how epigenetic changes can affect the evolution of tumor processes. So anyway, that was a schematic that just showed how, in order for a tumor mutation to propagate to future tumor populations, it would have to coincide with the permissive epigenetic state. Okay. So I guess we can just leave. So anywho, these epigenetic changes that occur. So in a, yeah, I guess this is just going through quickly. So I'll try to catch up with where the slides are. So the initial scripts, the initial schematic showed how epigenetic changes need to coincide with novel mutations in order for them to propagate to future tumor populations. And so when you have a situation where there's limited epigenetic changes, there's less opportunities for these mutations to propagate. But if you have higher levels of epigenetic disorder, you have a situation where the tumor is able to sample alternative tumor cell states in order to have a higher chance of clonal evolution. And so that was a general thing, but my research is focused specifically on glioma, which is the most common and malignant primary brain tumor. And this one is derived from precursors to intrinsic brain cells, such as oligodendrocytes and astrocytes, as shown in this top schematic here. Now, a lot of studies profiling tumors at the single cell level initially were based off of RNA sequencing. And so they looked at gene expression, and some of the results of these studies was looking at how, in this bottom schematic, you can see in this plot where each dot represents a cell, that these cells in glioma lie along this hierarchy that represents their stemness levels and also their recapitulation of glial cell lineages. And so this hierarchy is also dynamic. And interestingly, that one image is not showing right now, but there was a picture briefly that showed how this balance of cell states within a given tumor microenvironment can be affected by various external and genetic stimuli. Like, for example, radiation and chemotherapy, or hypoxia, which is a frequent feature of these more aggressive gliomas, as well as, say, extrachromosomal DNA, which the last presenter nicely showed an overview of. So for my projects, now that we've known that these gene expression changes can be shown and that you can see these differences in these cell states between different tumor micropopulations, in addition, we want to focus more on studying these regulatory mechanisms that result in these gene expression changes and what you see this hierarchy in. And so there's this recent platform that came out, which is a droplet-based sequencing technology that allows you to take a tumor tissue and then dissociate it into single cells and then simultaneously profile chromatin accessibility and gene expression. This chromatin accessibility is important because these open chromatin regions reflect active regulatory regions. And with this platform, you can directly link them to gene expression. So for my project, I'm applying this sequencing technique to the longitudinal profiling of glioma patients. So we take samples and then we sequence them at the primary tumor, and then we collect information on the treatments, and we can sequence them on the recurrent tumor as well. And so with this, we have this direct assessment of active regulatory regions and the gene expressions across time for multiple clinical time points in a given patient. So we can do a few things, like for instance, we can look at changes in gene expression as a result of treatment, and we can link it back to the specific, say, transcription factors or other key regulatory elements that are involved with these genetic changes. So we can look at cell state specific treatment mechanisms. We can also observe, for example, using the chromatin accessibility data, the presence of extra chromosomal DNA, and then link that to changes in oncogene enhancer activity as a result of it. So yeah, thank you. And for a static view of my work, you can come tomorrow to my poster.
Hello, everyone. I'm Ali Furobipour, and I'm a postdoc in Chung Lab at JAX. In collaboration with Wednesday, we have been developing an integrated deep learning model for stratifying colon adenocarcinoma patients. Colorectal cancers in general are the fourth most common cancer in the US, with an estimated 53,000 deaths in 2021. Its 5-year survival rate is around 65%, and its 3-year mortality rate has decreased from 23% in 1992 to about 13% in 2018. While this is good news, stratifying patients in the clinic remains a challenge. While guidelines and protocols for tumor boards are developed, there is still subjective variability between experts and may not efficiently capture all the complex relations between patients' data. Next, please. In this project, we integrate H&E whole slide images with clinical and genomic markers. The hope is that our models would learn the complex data patterns separating high-risk and low-risk patients. Since we're still analyzing images, we are using a deep learning pipeline, basically because deep learning is the only thing that works on images. In our pipeline, when a whole slide image comes in, we break it into smaller pieces called tiles. We only keep the tiles within the tumor regions and pass them through a convolutional neural network called Inception V3. Now, to do integrative analysis in a deep learning framework, we concatenate deep learning features with a panel of clinical and genomic markers selected by our collaborators based on their prior knowledge. All features are then fed to a multi-layer perceptron to distinguish tiles of high-risk and low-risk patients. So when a tile comes in, our neural network not only sees local information of the tile, but also contrasts them against the patient-level genomic and clinical information. Given the probability of being high-risk at the tile level, we can generate heat maps to visualize regions of interest or combine tile-level predictions to assign a risk score to the patients. Note we are using a classification-based model instead of the classical Cox model. This approach greatly improved our patient stratification. Next, please. Interestingly, when we use models trained only on images, we do not see a strong separation of patients in the KM plots, where blue is predicted low risk and orange is predicted high risk. We do not see a strong separation when training models only using genomic or clinical markers either. But as soon as we integrate them all, we see a strong separation. This suggests these data modalities complement each other and there's a strong crosstalk between them. So you only have enough information when you use all data modalities. We are now trying to quantify and identify the important crosstalks. For instance, we have noticed the crosstalk between image and clinical features is important for 3-year survival prediction, and the crosstalk of image features and genomic markers are more important for long-term survival analysis. Next, please. So in conclusion, integrative analysis, in other words, combining data of different modalities, is important for survival analysis and patient stratification. Our analysis further confirms that different data modalities complement each other, and I'd like to emphasize how important it is to study the crosstalk between them, as they pack a lot of information. To that end, deep learning is an efficient technique for such tasks. We will be further deconvolving the crosstalk, validating our model on external data sets, and extending our integrative analysis pipeline to other cancers. If you're interested in our work, come see my poster tomorrow at poster group 7. Finally, I would like to thank everyone in Chuang Lab, particularly Jie Zhu, who is a very talented graduate student who did most of the coding, and our collaborators and Wayne State. And thank you all for listening. That's it.
Excellent. All right. So this concludes our postdoc Speak for Science event for 2021. I want to thank all of our postdoc speakers this afternoon for doing such a great job and sharing their work with us. I enjoy this day so much every year to see these little snippets of all the really important work that's going on here on our Farmington campus. And so again, I thank our postdocs for doing that for us and sharing this work. I want to thank all of you in the audience for joining us this afternoon and invite you back tomorrow at 3 o'clock for our poster sessions, where again, you can hear all of our speakers today in more detail and kind of dive into the weeds with them on their work and ask them the questions that maybe you didn't get to ask today. And there'll be several other postdocs that'll have posters as well that you didn't get to hear from today. So that should be a really fun event. So please come back. Thank you again. Have a wonderful afternoon and hope to see you tomorrow.